Dear students, today we will learn about brief outline of cancer biochemistry and cancer phase of cell cycle. First of all, let us see what is cancer. Cancer is a disease associated with uncontrolled proliferation of cells. Change in nucleic acid sequences coding cell cycle regulatory proteins lose their respective functions that lead to uncontrolled proliferation of cells. Proto-oncogenes and tumor suppressor genes are two such nucleic acid sequences. The gain of function of proto-oncogenes and loss of function of tumor suppressor genes caused by chemical and physical carcinogens and tumor viruses led to the onset of tumors. A dividing cell like cancer cell requires three basic needs: rapid ATP generation to maintain energy status, increased biosynthesis of macromolecules, and tightened maintenance of appropriate cellular redox status. To meet these needs, cancer cells acquire alterations in the metabolism of all four major classes of macromolecules: carbohydrates, proteins, lipids, and nucleic acids, which are done in a stressful and dynamic microenvironment of solid tumor where concentrations of crucial nutrients such as glucose, glutamine, and oxygen are spatially and temporally heterogeneous. Now let us see few characteristics of cancer cells. Rapid ATP generation in cancer cells. Tumor cells must generate the energy to support rapid cell division. The best characterized metabolic phenotype observed in tumor cells is the Warburg effect, which is a shift from ATP generation through oxidative phosphorylation to ATP generation through glycolysis that provide ATP more rapidly even under normal oxygen concentrations. Many transformed cells derive a substantial amount of their energy from aerobic glycolysis, converting most incoming glucose to lactate rather than metabolizing it in the mitochondria through oxidative phosphorylation. As a result, tumor cells take up abnormally high rate of glucose. It is found that the shift of aerobic glycolysis by cancer cells is due to the activation of oncogenes, loss of tumor suppressor genes, and upregulation of P13K pathway. In addition to pyruvate derived from glycolysis, fatty acids and amino acids glutamine, BCAAs, isoleucine, valine, and leucine generate acetyl-CoA and other organic molecules that enter TCA cycle for the production of mitochondrial ATP. Their mitochondria can function optimally even at oxygen levels as low as 0.5%. To survive the hypoxic tumor microenvironment, cancer cells diminish their demand for ATP by decreasing high ATP-dependent processes such as running the sodium-potassium-dependent adenosine triphosphatase. If diminishing ATP demand is not sufficient to maintain ATP-ADP ratio, the rise in ADP activates adenylate kinase and converts two ATP molecules into adenosine 5-monophosphate AMP and ATP. In an environment where nutrient supply is poor, tumor cells adapt to the microenvironment by decreasing their demand for ATP. The rise in AMP during nutrient deprivation also triggers the activation of AMP kinase, AMPK, which activates catabolic pathways like fatty acid oxidation to stimulate ATP production. The resultant increase in ATP availability maintains an adequate ATP-ADP ratio to drive unfavorable biological reactions. The next point is increased biosynthesis of biomolecules. In cancer cells, the biosynthesis of macromolecules such as proteins, lipids, and nucleic acids is enhanced via tumorigenic mutations, particularly P13-mTOR signaling. Under the influence of growth factors, cancer cells express surface transporters that allow them to acquire amino acids from the extracellular space. This not only provides cell with the raw materials needed for protein synthesis, but also allows them to maintain activity of the mTOR signaling system. mTORC1, which is a part of mTOR signaling system, stimulates the uptake of glutamine and its conversion to glutamate by glutaminase. The excess glutamine is exported from the cell in exchange of essential amino acids. Thus, growth conditions with abundant essential amino acids and glutamine activate mTORC1 mediated protein synthesis. When nutrients are scarce, cancer cells employ a number of pathways to supply key pools of intracellular metabolic intermediates. Intracellular proteins are recycled through autophagy to supply amino acids. Extracellular proteins and other components are internalized by macropinocytosis to supply nitrogen and carbon to central metabolic pathway. Fatty acids are rapidly produced in cancer cells. Acetyl-CoA, which is required for the fatty acid synthesis, is derived from many sources, including glucose and leucine. Glutamine and acetate also provide carbon sources when access to glucose-derived acetyl-CoA are impaired. Transcription factor SREBP1 regulates fatty acid synthesis by regulating enzymes needed to convert acetyl-CoA into fatty acids, as well as the pathways required to convert acetate and glutamine into acetyl-CoA. In cancer cells, SREBP1 is kept transcriptionally active by mTORC1 signaling. The lipogenesis in proliferating cells are also maximized by acquiring fatty acids and lipids from extracellular space, as well as suppressing its oxidation through P13K signaling. The next characteristic is the tightened maintenance of appropriate cellular redox status. Cancer cells have an increased rate of specially localized mitochondria and NADPH oxidase NOx dependent reactive oxygen species production due to a combination of oncogenic lesions and tumor microenvironment. This allows for the proximal activation of signaling pathways of P13K and mitogen-activated protein kinase, extracellular signaling, regulated kinase, MAPK-ERK, and transcription factors, HIF and nuclear factor kappa B and F kappa B, necessary for tumorigenesis. However, as the hydrogen peroxide ROS levels increase, cell death signaling pathway is initiated. Hydrogen peroxide is converted to hydroxyl radical, which can directly damage DNA proteins and lipids. Thus, the localized ROS in cancer cells, which activate signaling pathways and transcription factors to promote tumorigenesis, need to be buffered from reaching levels of ROS that incur cellular damage by the increased expression of antioxidant proteins. This is done by producing higher levels of ROS scavenging enzymes. One mechanism by which cancer cells increase their antioxidant capacity is by activating a transcription factor called nuclear factor related factor 2. The activated NRF2 induces transcription of many antioxidant proteins, including GPXs, TXNS, as well as enzymes involved in GSH synthesis and cysteine import through the cysteine glutamate antiporter. NRF2 also regulates the serine biosynthesis pathway, generating NADPH in the mitochondria, which is required to maintain the antioxidant capacity of GPXs and TXNS, and critical for redox balance under hypoxic conditions. Now, coming to the cancer phases of cell cycle. Regulatory proteins assure the proper coordination of cellular growth during G1 and G2 phases, DNA synthesis during the S phase, and chromosome segregation during mitosis. The cells that have DNA damages are arrested for further progress in the cell cycle till their DNA is repaired or they commit suicide by a programmed cell death. Two genes, proto-oncogenes and tumor suppressor genes, play a key role in the cell cycle regulation. Mutations in these two genes results the loss of cell cycle regulation and onset of cancer. Now there are some restriction point control and progression to S phase from G1. The regulation of transition of cell cycle from G1 to S phase is very important for the onset of cancer. In normal cells, presence of enough mitogen stimulations is required for the progression to S phase. Otherwise, the cell withdraws from the cycle into a resting state called the restriction point. Cancer cells abandon these controls and tend to remain in cycle. They also avoid maturation and terminal differentiation of cells. The decision to divide occurs as cells pass the restriction point let in G1, after which they become refractory to extracellular growth and regulatory signals and instead commit to the autonomous program that carries them through to division. The progression of cell cycle from G1 to S phase is controlled among other factors by tumor suppressor gene RB. PRB is deployed by the cell cycle clock to serve as a guardian of the restriction point gate. In early G1, PRB is found in hypophosphorylated state and tightly binds and represses the activity of the E2F family of transcription factors which are functionally required for expression of genes necessary for S phase. Under normal circumstances, phosphorylation of PRB is initiated midway through G1 by active cyclin D CDK4 and cyclin D CDK6 complexes. The expression of D-type cyclin gene is induced by mitogens. PRB phosphorylation is completed by other cyclin CDK complexes in late G1, allowing activation of E2F transcription factors which stimulate transcription of genes encoding proteins required for DNA synthesis. The complete phosphorylation of PRB irreversibly commits the cell to DNA synthesis and the cell progresses to S phases. In absence of mitogens, cells cannot synthesize D-type cyclins. PRB will remain in dephosphorylated state and the cell cycle will halt at G1. If the function of PRB is lost, then it cannot regulate the transition of G1 to S phase through restriction point. The loss of function of PRB can be done through mutation of the chromosomal RB gene copies, methylation of RB gene promoter, or the actions of DNA tumor virus oncoproteins. In some cases, PRB phosphorylation is deregulated, resulting in inappropriately phosphorylated and functionally inactivated PRB. In other cases, dephosphorylation of PRB, which normally occur at mG1 transition through the action of protein phosphatase enzyme, never occur, leaving PRB in its hyperphosphorylated inactivated state throughout the entire growth and division cycle of the cells. The oncogenic mutations of proto-oncogenes can also inactivate the PRB functions. Without a functional PRB, cells move from G1 to S phase even in the absence of mitogens or without repairing the damaged DNA or without being subject to the usual controls that are designed to ensure that cell cycle advance can proceed only when certain preconditions are satisfied. The p53 protein is a sensor essential for the checkpoint control that arrests the cells with damaged DNA in G1. p53 mutations abolish its ability to bind to specific DNA sequences and activate gene expression. Cells with functional p53 become arrested in G1 when exposed to DNA-damaging irradiation, whereas cells lacking functional p53 do not. p53 protein is highly unstable and present at a very low concentration in normal cells. Their concentration heightened only in stressful situation when activated ATM kinase phosphorylate and stabilized them. The stabilized p53 activates the expression of p21 CIP gene, whose product binds to G1 cyclin CDK complexes and arrests the cell. When the p53 G1 checkpoint control does not operate properly, damaged DNA can replicate, perpetuating mutations, and DNA rearrangements are passed on to daughter cells, contributing to the likelihood of transformation into metastatic cells. p53 also induces the arrest of cells in G2 M phase transition when DNA is damaged following replication. p21 CIP and two other proteins induced by p53 inhibit cyclin B CDK1 complexes required for entry into mitosis. p53 also represses expression of the genes encoding cyclin B and topoisomerase II, which are also required for G2 mitosis transition. If DNA is damaged following its replication, p53-induced G2 arrest will prevent its transmission to daughter cells. Now, finally coming to the conclusion, mutations in tumor suppressor genes and proto-oncogenes initiate tumors and their progression required many biochemical changes. The cancer cells are able to adapt a stressful microenvironment where crucial nutrients and oxygen supply are scarce and heterogeneous. They can generate more ATP from different sources and at different environmental conditions, even under hypoxic conditions. The synthesis of macromolecules, proteins, lipids, and nucleic acids required for the rapid growth is also enhanced through different mechanisms like tumorigenic mutations that activate various signaling pathways. The supply of key intracellular metabolic intermediate is also enhanced. With the progression of cancer, production of reactive oxygen species are also increased, which is detrimental in the survival of the cells. This increased ROS is countered by increasing the production of antioxidants like GPX and TXNs. Progression of cells from G1 to S phase is very crucial for the development of tumor. Normally, this progression is done if growth factors are present and enough mitogen stimulation is provided before restriction point. Withdrawal of mitogens before the restriction point results in the exit of the cell from the cycle. Tumor suppressor genes PRB and p53 play crucial roles in the progression of the cells from G1 to S phase. Functional PRB and p53 inhibit the progression of cells through restriction point in the absence of enough mitogen stimulations and DNA damages. Mutations in PRB and p53 genes results in the unrestricted progression of cells from G1 to S phase even in the absence of mitogens or damaged DNA. p53 also check the progression of cycle to M phase if DNA is damaged following replication.